Hello everyone. Today we will learn about how to check quality of a FastQ file. In the FastQ tutorial, we have learned that FastQ is the most common file format to store raw sequencing information for most of the sequencers. We also learned that FastQ files not only contain the DNA sequencing information but also stores the quality of each of the bases. That means some nucleotide bases have higher quality and some have lower qualities. The raw sequencing reads or the sequences may contain contaminations such as adapters and other DNA sequences. Through checking the quality of a sequencing library, we will assess the quality of nucleotide bases as well as the presence of any contaminants. For that, we will be using FastQC, which is one of the most popular tool to assess the quality of a FastQ file. So to start, we will install the FastQC software into our working environment. We'll go to Anaconda page and then copy this line, and then we will install it into our working environment. So now we will paste this line to install FastQC. Yes. Yes, the installation was successful. So now in this directory there are two FastQ files. This is a paired in library. Now we will check the quality of both files. The command is very simple. We need to specify the file name. That's all. And it will generate the report for us. FastQC has generated two outputs. One is an HTML file, one is a zip file. Zip file contains the raw information, but we will open the HTML file to export the data. Also, I want to show another command. We can run multiple FastQ files in a single command. FastQC, if we use the wildcard command, this will run for all the FastQ files within this directory. Now we can check for both FastQ files. The output for FastQC has been generated. So now let's take a look into the HTML file. This is how the FastQC output looks like. There are several summaries and that tells about the quality and overall statistics. Also, there are some details about per base sequence content, GC content, presence of N bases, duplication level, whether sequences are overrepresented or not, and also about the information for adapters. In FastQC report, this tick sign is used to indicate that passes the quality, and cross signs are used to indicate the failure. So when there is a cross sign, that means we need to investigate further what is happening in this library. The basic statistics here is telling the file name, file type, encoding, total number of sequences, sequence length, etc. The par base quality score is very important for us. We need to check whether some bases have low quality on average in this library. So generally Illumina library has poor quality bases at the 3' end. So for this library we can see most of the reads are above Q20. So we can also use Q30 as our cutoff to ensure that all the bases have higher quality. So let's take another score, per sequence quality score. This is very similar to the previous plotting. Most of the sequences have quality score which is above 20. Let's take a look into the GC content. First QC is suggesting a failure here. The reason is we can see our library is deviated from the theoretical distribution for GC content. Sequence length distribution is okay, and sequence duplication level does not look okay, because we can see in this library there are a lot of reads that are duplicated. A diverse library should have a lower level of duplicated reads. A higher duplication level indicates certain biases such as PCR amplification bias. This library here we are showing is an PCR amplicon library. Probably for that reason, there is a higher level of duplications. Overrepresented sequences, if a sequence is present more than 0.01% of the library, those are defined as overrepresented sequences. And in this library, we can see there are some overrepresented sequences. FastQC can detect some of the known or common adapter sequences in a library. In this case, it is showing there is no common adapters present in this library. But we need to be aware of the fact that since it is not reporting any adapter, it does not mean our library does not contain any adapter. So we need to contact to the sequencing lab. What is the adapter sequence? Then we can identify the presence of those adapters in our library. So in summary, we have learned how to install FastQC software and also how to run FastQC to generate those statistics that are essential to understand the quality of a sequencing library. In the next tutorial, we will learn about how to remove the low quality bases from a library. Also, we will learn about how to remove adapter sequences. Thank you for watching this video. See you in the next one.